Hello students. Today we are here to learn the production of the bacterial and the non-bacterial toxins in food, which ultimately leads to food poisoning. Now what is food? Food is basically a source of nutrient for the human, for the animals and for all the biological entities on this earth. And therefore, food is a very good substrate for the growth of microorganisms like bacteria and mold. Therefore, the food has to be consumed by the human and the food is also consumed by the bacterial and non-bacterial species. And when these bacterial and non-bacterial species grow upon the food, secrete some substances which are known as toxin, makes the food poison. So let us learn who causes this food poisoning and what happens to these poisoning processes. We begin our learning with the bacterial toxins. Based on the structure and the release of the toxin, the bacterial toxins are categorized into two types. One is called the endotoxin and the other one is called the exotoxin. The endotoxin is the one which is an integral part of the bacterial cell which is not released in the external environment. For example, the lipopolysaccharide of the gram-negative bacteria. However, the exotoxins are the proteins or the substances which are released by the bacterial cells in the external environment, and therefore this is mainly seen in the case of gram-positive bacteria. So on the other hand, we can say that endotoxin is a characteristic property of gram-negative bacteria, while exotoxin is a characteristic property of gram-positive bacteria. However, there are many gram-negative bacteria which produces exotoxins and release them into the external environment. The exotoxins, which are proteinaceous in nature, mimics the characteristic feature of the enzymes. Number one, they are protein in nature. Number two, they have got a specific activity. Number three, they have specific substrates or we say site of action upon which they act. And number four, they are potent in a very minute amount. So these exotoxins based on the site of action can further be categorized into three important categories, and they are enterotoxin, neurotoxin and cytotoxin. Enterotoxin refers to the toxin which act on the enterocytes, that means the cells of the gastrointestinal tract. On the other hand, neurotoxin refers to the toxin which acts on the nerve cells or neurons and therefore called the neurotoxins. The cytotoxin, on the other hand, refers to the toxin where they act on specific cell types only, and therefore the name comes cyto, meaning cell. So what is enterotoxicosis? Enterotoxicosis therefore refers to the true food poisoning which is caused by the ingestion of a food which is already contaminated by a toxin or by the presence of a bacterial toxin. So the bacterial species which cause this food poisoning are as follows. They can be Clostridium botulinum, Staphylococcus aureus, Clostridium perfringens, Salmonella typhimurium, Bacillus cereus, Escherichia coli, Yersinia enterocolitica and so on. So let's begin with the first organism that is commonly known to us is Clostridium botulinum. Now this Clostridium botulinum causes food poisoning and this is referred to as botulism. This is due to release of an enterotoxin which affects the gastrointestinal tract of the human body. Now this botulinum toxin which is released from the Clostridium botulinum affects or is mainly found in the home canned food which are not properly preserved and is also being found in the smoked fishes. The symptoms commonly encountered by this type of food poisoning is vomiting, diarrhea, gastric distension, muscular weakness, cramps, constipation, difficulty in urination, dry and rough surface tongue Along with it, we have a very prominent symptom which is progressive respiratory paralysis. Now, why suddenly the paralysis takes place along with the food poisoning? This happens because botulinum along with the enterotoxin is also a neurotoxin which affects the nerve cells. Now, how does it affect the nerve cells? Let us learn this. We know that we have got a neuromuscular junction where the nerve cells come in contact with the muscles. Now, this gap which is there between the nerve cells and the muscular cell or the muscle cell is called the synaptic cleft. So, in this synaptic cleft, the release of neurotransmitter is very essential because through this neurotransmitters, the nerve sends the signal to the muscles and therefore the muscles contract or the muscles relax depending upon the neurotransmitter that is being released. Now, if I go into the detail of this, let us first study what happens in the normal functioning at this neuromuscular junction. We find that inside the neurons there is presence of synaptic vesicles. Now, these synaptic vesicles store some granules which are called neurotransmitters and they are acetylcholine. This acetylcholine has to be released in this synaptic cleft so that it reaches the receptor and the muscles will contract. This release of acetylcholine takes place with the help of the snare proteins which are synaptobrevin associated with the synaptic vesicle and syntaxin and SNAP25 which are associated with the inner membrane. Now, this SNAP25 and syntaxin gets associated with the synaptic brevin. As a result, the vesicle comes down towards the inner surface and ultimately the release of acetylcholine takes place. Now, this is the normal transmission. However, when the neurotoxin, that is botulinum toxin, attacks, what happens is botulinum has got a bipartite structure. That means it has got an A subunit and a B subunit. The B subunit is the one which binds and the A subunit is the virulent part of the toxin. So, through receptor-mediated endocytosis, this toxin, the B subunit binds and the A subunit ultimately is released and this comes out and ultimately causes the cleavage of the snare proteins. That means syntaxin, SNAP25 and synaptobrevin. They are all lost. When they are lost, what happens is this vesicle which stores the acetylcholine will not come down towards the inner membrane and therefore no release of the acetylcholine takes place and the muscles which is present over here will not contract. So, if this toxin is present in the body, there is no contraction of the muscle, ultimately this leads to flaccid paralysis, and that is what happens in the respiratory paralysis or whenever we consume the Clostridium botulinum spores along with our food item. We come to another species which is Clostridium perfringens. Now, this Clostridium perfringens again releases an enterotoxin which is released along with the sporulating cells. That means before the lysis of the cell's sporangia, along with the spores, these enterotoxin which are released by perfringens is released into the body. This is mainly found in contaminated meat, poultry dishes and then sausages. We find that cramps and diarrhea within 12 to 24 hours appear. However, there is no vomiting and no fever. The disease called the sudden infant death syndrome or SIDS is a very common symptom for this type of enterotoxin poisoning. Now, in sudden infant death syndrome, the death of the infants or small children takes place because the small children are regularly given honey. Now, this honey is a very good source or a very good media for the growth of Clostridium perfringens. So, when honey is taken in by the children, the spores are getting inside the gastrointestinal tract which ultimately germinate, mature and during sporulation, they release this enterotoxin. When this enterotoxin is released in the body of the child ultimately is subjected to the paralysis, the suffocation takes place and the child suddenly dies and therefore this leads to SIDS. Staphylococcus aureus is another genus which is responsible for food poisoning, and it releases an enterotoxin which is called Staphylococcus enterotoxin. Along with it, it also releases a toxin which is called TSST, that is Toxic Shock Syndrome Toxin. Now, this toxin is called a superantigen. Why superantigen? Because this toxin can bind to the receptors of the T cell, the cells which participate for our defense mechanism. When they attack the T cells, there is release of interleukins, gamma interferon and tumor necrosis factor, which ultimately leads to gastroenteritis. Now, this contamination of Staphylococcus aureus can be found in meat, seafood salads, sandwich spreads and high salty foods, and therefore it causes the poisoning of the food. The symptoms are normally nausea, vomiting, diarrhea within 4 to 6 hours. However, there is no fever in this case. Salmonella typhimurium is another gram-negative bacteria which causes food poisoning. Though it is gram-negative, it also releases an enterotoxin which is called the salmonella toxin. Now, this salmonella toxin, it activates the adenylate cyclase. When adenylate cyclase is activated in a cell, there is a loss of electrolytes along with water, and as a result, this causes watery diarrhea. The high protein food is also seen to be contaminated or poisoned, like the meat, poultry, fish and eggs. The common symptoms with salmonella is seen to be diarrhea, nausea, chills, vomiting and fever within 12 to 24 hours of consumption of this type of contaminated food. Salmonella typhi is another organism which does not release any toxin. However, this may contaminate the food, especially the fruit juices or any liquid item which is mixed with some water. Now, this water may contain Salmonella typhi, which ultimately leads to a disease commonly known to us as typhoid. The Salmonella typhi, which is a gram-negative bacteria, enters the M cells. The M cells are present in the Peyer's patch of the gastrointestinal tract. Through this M cell, the Salmonella typhi enters, reaches the macrophages which are present in the lymphoid tissue, and after reaching the macrophages, it reaches the mesenteric lymph nodes, and via the bloodstream, it reaches the spleen and the liver. From the liver, via gallbladder, it can again attack the gastrointestinal tract and cause the necrosis of the Peyer's patch. So, we can see that how salmonella poisoning can affect the different cell types in a human body. Coming on to another genus which is again a gram-positive bacteria is Bacillus cereus. Now, this Bacillus cereus causes gastroenteritis which is of two types, or basically it shows two prominent symptoms. One is emetic and the second one is diarrheal. Emetic means you have acute vomiting, and diarrheal types means you have symptoms of acute watery diarrhea. This is because of the presence of the extracellular toxins and enzymes which are released by Bacillus cereus. The different type of enzymes that are released by Bacillus cereus is lecithinase, protease, beta-lactamase, sphingomyelinase, cereolysin and then hemolysin. Bacillus cereus poisoning is mainly seen to be present in the starchy food. Mild cases of diarrhea and some nausea may develop within 12 to 24 hours. Escherichia coli, commonly known to us as E. coli, is present in the human body. So, how it can be harmful? Yes, it is harmful because there are many strains of Escherichia coli which are very much dreadful and pathogenic, like EPEC, which is enteropathogenic E. coli, EHEC, which is enterohemorrhagic E. coli, ETEC, which is enterotoxigenic E. coli. All these strains produce the enterotoxin and therefore leads to watery diarrhea and the symptoms of vomiting. The two toxin which is produced by Escherichia coli is STX1 and STX2, which inhibits the protein synthesis of the endothelial cells, because the endothelial cells has got presence of receptors which is called GB3, which is nothing but globotriacyl ceramide. When they bind to GB3, as a result of this binding, the cells cannot perform the function of protein synthesis and they die. STX1 and STX2 are heat-stable toxin. This is mainly found in the food poisoning caused by meat and cheese. The symptoms of abdominal cramps and diarrhea appears. However, there is no symptom of fever. Shigella dysentery is another organism which leads to food poisoning. Now, Shigella, though is a gram-negative, it produces an exotoxin which is Shiga toxin. This Shiga toxin can bind to the GB3 receptors of the endothelial cells. That means the endothelial cells are mainly found in the vasculature of the kidney, and therefore this Shigella dysentery poisoning leads to a symptom which is called HUS, which is hemolytic uremic syndrome. The vasculature of the kidney has got presence of endothelial cells. Small blood vessels are present, and on the surface of these blood vessels there is presence of receptors which is GB3. Shiga toxin comes and binds to this GB3 and as a result causes the death of these cells and the blood is being released. Along with this, therefore, we find the bloody diarrhea, the contaminated sprouts, the seeds, the unpasteurized milk and the apple cider may show presence of Shigella dysentery. Vibrio parahemolyticus is another bacterial species which is found in the seafoods and causes the food poisoning. Here we find that the raw and the cooked seafood may show the presence of this type of Vibrio parahemolyticus. We have the symptoms of diarrhea, cramps, vomiting, headache and fever which can last for many hours. However, the symptoms appear within 12 to 24 hours. Listeria monocytogenes. Now, Listeria monocytogenes is a bacteria which causes a disease called listeriosis. Now, a special characteristic feature of Listeria is they are able to survive at very adverse conditions. They can tolerate the refrigerated temperatures. They can tolerate a pH range which is from 5 to 9.5. So, from an acidic pH to highly alkaline pH, they can tolerate this range. The salt tolerant capacity is also there. For example, they can tolerate a percentage of 30.5% for 100 days if the temperature is 39.2 degrees Celsius, which is quite low. However, they can survive for only 5 days if they are held at 98.6 degrees. So, basically, they are susceptible to higher temperature. However, they are resistant to lower temperature. So, frozen foods are mainly contaminated with this Listeria monocytogenes. The frozen food like the milk, cheese, vegetables also. Now, this food poisoning mimics the symptoms of meningitis and this is also found to be susceptible in the immunocompromised individuals. Listeria monocytogenes, when present in the contaminated food, comes into the intestine of the human and through lymph node reaches the bloodstream. When it comes to the bloodstream, it may affect the liver or it may affect the spleen. Along with it, it may also affect the brain and therefore leading to meningitis. However, if this food poisoning is taking place inside a pregnant mother, it can also pass through the placenta and reach the fetus and therefore the fetus is also affected by this type of food poisoning. Yersinia enterocolitica is another bacterial species which produces the food poisoning and therefore referred to as gastroenteritis. It can grow at the refrigerated temperature and the common symptom is diarrhea, abdominal pain, vomiting. It mimics the symptoms of appendicitis and therefore it has been found in the medical history that many patients undergo the operation of appendix. However, they were suffering from Yersinia enterocolitica. Yersinia enterocolitica, like the Shiga toxin and the Salmonella, enters inside the human intestine through the M cells and ultimately reaching to the macrophages which is present in the lymphoid tissue. They destroy the macrophages by forming the pore or by inhibiting the phagocytosis and then by inhibiting the TNF production. Ultimately, the macrophages die and the cells are released which again enter the epithelial cells or the cells of the gastrointestinal tract, and they invade the epithelial cells, and therefore the whole lining of the gastrointestinal tract is being affected. Coming on to the second type of toxin, which is of non-bacterial origin. If they are non-bacterial, then who are the one who are producing it? They are basically produced by the fungus, and therefore they are referred to as the mycotoxins. The important mycotoxin that is known to cause food poisoning is aflatoxin. Aflatoxin is very much poisonous and deleterious, and it is produced by Aspergillus flavus, Aspergillus parasiticus and Aspergillus nomeus. It is found to be present in stored wheat, corn, cornseed, rice, barley. So any pulses or the grains which can be stored for a much longer time in humid condition can lead to food poisoning. Chemically, aflatoxin is having a structure which is bisulfuran polycyclic. Because of this structure, when they are exposed to the ultraviolet light, they give fluorescence. Here you can see that the fluorescence which is given by aflatoxin G1 and G2, which is green in color. This is under normal light and this is under ultraviolet light. However, the aflatoxin B1 and B2 produce the blue fluorescence. This is the blue fluorescence seen over here, and this is under the white light. So because of this fluorescence, it becomes very helpful for the detection of the particular type of the mycotoxin that has affected the food item. This type of toxin basically affects the liver of the human body. Coming on to the second type of the mycotoxin, and that is called ochratoxin A. Now this ochratoxin A is produced by Aspergillus ocraceus and Penicillium verrucosum. This is found again in corn, wheat, barley, rice, oats, coffee beans, and possibly we say that this toxin is responsible for a disease which is known as the Balkan endemic nephropathy. Ochratoxin is seen to be more stable in humid condition in contrast to the aflatoxin B1. Patulin is another type of mycotoxin that is produced by many species of Penicillium, Aspergillus and Bisoclamis. They again affect the grain, the cheek starter, malt feed, moldy bread, bakery goods. So all these items which you see over here can be affected by the presence of this patulin. Here you can see the picture of an infected apple juice. Now this toxin is highly reactive because it binds to the sulfhydryl group which is present in cysteine, thioglycolic acid and glutathione, and therefore the metabolic pathway of the human cells is being affected. Coming on to another mycotoxin, that is citrinine, produced by the Penicillium citrinum and Penicillium viridicum. This is usually accompanied by the production of ochratoxin A. Now this is found in mixed barley, oats, corn or peanut. Here you can see the picture of the corn being affected. This basically affects the kidney of the human body. Fumonisin is another mycotoxin that is released by Fusarium moniliforme. It is seen that about 80 to 100% of the corn that is being harvested is affected by fumonisin. Now this causes the inhibition of the de novo synthesis of the sphygmocin. T2 toxin is the mycotoxin which is produced by many species of Fusarium. This toxin is basically seen to be causing the food poisoning of the millet, wheat, rice, oats, and it is an inhibitor of the protein and DNA synthesis in the mammalian cells. So this is very potent and very poisonous for the humans to consume this type of food item which is affected by T2 toxin. So we can conclude that the food is a very good source of nutrient for our growth and survivability. But the food we are consuming should be safe. It should be free of toxin. It should be free of any microbe. Because the microbe which helps in the production of the food is a useful microbe. But the microbe which is present after the food is already being produced is not a good microbe, but it is a harmful microbe. Therefore, the food when consumed should be well preserved, well canned, well heated, well cooked in order to see that no toxin is there and no bacterial species or fungal species is there. Thank you.